next week in lab, please take note of what's on the board. We're doing the P-globe bacterial transformation. Before you come to lab, be sure to read page 1 and complete lesson 1 on page 2. There will be a quiz on the material on pages 1 and 4.
You will be collecting the first one when they walk into lab, right?
Yes.
Okay.
No, because the backside has to be completed as well before. But I can check.
Okay. And then your reading quiz Monday, remember, 13.1 and 13.2. So that will carry us through information that we're covering today. And then I think we're coming up with, isn't it a week from Monday? Another exam? So that will be on photosynthesis, DNA replication, and part of chapter 17. We'll see how far we get. My intention is to try to wrap stuff up on maybe Tuesday, Thursday at the latest, so there'll be at least a day break in between. Give us a chance to do some review on Friday. Things are getting hectic. Okay, anybody have any questions? Hopefully right now you should be quite comfortable with basic structure of DNA, and particularly the two most important features I think are understanding the base pairing rules, the A and T base pair and G and C base pair, and in this anti-parallel nature of DNA, which end of a DNA chain terminates in a phosphate group, the 5' end or the 3' end? 5' end, okay. 5' end terminates in a phosphate, 3' end terminates in a hydroxyl group, which is on that deoxyribose sugar. So if one strand is that oriented 5' to 3', the other one is opposite that orientation, head to tail, tail to head. Well, that's a problem. At least it's a problem in terms of the original conception about how DNA replicated. The idea was right that the two strands were templates to two newly synthesized strands, meaning that replication was semi-conservative. But the idea that was envisioned, especially by Watson and Crick, initially was the fact that the DNA just unwound from one end, and that both strands were copied in the same direction as the molecule continued to unwind. And eventually it completely unwound, both parent strands were copied, and now we had two DNA daughter molecules. The problem with that is the two parent strands are anti-parallel, and the enzyme, what's the enzyme that copies DNA? DNA polymerase, okay, only works in one direction. It only works in its 5' to 3' direction. So there's got to be a hydroxyl group sitting there to attach the next nucleotide in the growing strand. Well, if that's the case, if that enzyme only works in one direction, but the two strands are anti-parallel, then you can't just copy both from one end to the other. It just doesn't work that way. Okay? So, one of the questions is there another polymerase? Is there a polymerase that works in the opposite direction, in 3' to 5'? And as it turns out, no one's ever discovered one. And one of the things I like about science is you always learn something new. And I was doing some digging around yesterday saying, why isn't there a 3' to 5' polymerase? And actually I found a reason for that. I'm not going to go through the detail of that right now. If this is really a burning question in your mind, stop by sometime and I'll explain it to you. I know Dr. Morgan will be talking to me after class. He's interested in that kind of stuff. But no, there's only one kind of enzyme. There's only a 5' to 3' polymerase. So, how do we copy DNA given that particular limitation? Well, there's going to have to be different kinds of replication occurring on the two strands. So we have what we call a leading strand, which carries out continuous or uninterrupted replication. I like that word, uninterrupted. And a lagging strand where we have discontinuous replication, which is interrupted because as the DNA helix separates one of the strands, you've got to wait for the replication fork to open up before you can then copy backwards away from that replication fork. So one strand is being copied moving toward the opening replication fork, and one is being copied moving away from it. And so that one is interrupted. So that little fragments of DNA are formed as that interrupted DNA synthesis occurs. We call those Okazaki fragments. And eventually those Okazaki fragments are going to have to be joined together to make a continuous piece of DNA. So what's that look like? Okay, well, let's show you some static images first, and once we walk through this, I'll show you a couple of YouTube videos which give you a little bit more three-dimensional representation. So here's our replication fork right here. Again, dark blue represent parental DNA strands. You see the 5' to 3' direction, 3' to 5' direction of those two parental strands. And remember that the new strands that are made also have to have this anti-parallel relationship. So if this is this strand right here going 5' to 3', the newly made strand is going to have to run 3' to 5'. Okay? So we've got these enzymes that start breaking hydrogen bonds and separating the parental strands, and we start exposing the template for copying. And on this particular strand right here, the leading strand, once you put in that primer, and what kind of molecule is the primer, remember? RNA, okay? And the enzyme that adds that primer is called RNA primase. So once you start putting in that primer and get a few nucleotides fixed into that position, then DNA polymerase can come and just extend. And every one of these nucleotides that's been added has a hydroxyl group sitting on its end. So that means that a new nucleotide can bond together by its phosphate group joining to the hydroxyl group. And this can just continue uninterrupted as that replication fork proceeds toward the end of the molecule as it unzips. But on this other strand, the lagging strand, we can't synthesize in this direction because that's 3' to 5'. So we have to backfill and synthesize this direction. So that requires, when this is opened up to this point, a primer can be produced, and then we can lengthen back this direction. When the fork is opened a little bit more, another primer can be produced, and then we fill back in this direction. And then again here you can see that it's opened up a little bit more, another primer. So we make all these little short segments of DNA, which eventually are going to have to be stitched together by DNA ligase. So this represents the leading strand with continuous replication, and this represents the lagging strand. And then there's just a set of individual images that shows you what's happening on the lagging strand. So our replication fork has exposed some DNA. We form a primer first of RNA. That primer can then be extended by DNA polymerase. And when you reach the end of the molecule, we've got this Okazaki fragment. But now, as the replication fork exposes more template, another primer is going to have to start. More extension by DNA polymerase. Eventually, this primer is going to have to be removed, and these Okazaki fragments will have to be joined together to give us our complete piece of DNA that's formed on that lagging strand. Okay? So, DNA replication isn't quite as straightforward as we had originally envisioned. And it's made even more complicated by the fact that replication of DNA actually occurs in two directions away from the origin of replication. And so this gives you a good summary kind of some of the enzymes that are involved and what's happening in terms of replication on the leading strand versus the lagging strand. And here are some enzymes that are required for synthesis both on the leading strand and the lagging strand. And we see a lot of overlap between those particular enzymes. But it's important to understand that both of these processes have to occur to duplicate your entire DNA molecule. Okay, so let's take a look at a couple of videos and see if this makes a little bit more sense to see it in animation.
begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Don't worry about this. I think the previous illustration that they gave showing kind of two polymerases moving kind of opposite directions is fine for your understanding. In reality, we believe that there is a complex of DNA polymerase molecules, and because this particular complex bends this lagging strand of DNA backwards, then the complex can move in one direction, but we're still effectively having a leading strand and a lagging strand. But the way this DNA is looped back on itself makes it possible for this complex to move toward the replication fork. There's another video that shows you a little bit more detail than that, and I'm going to show you that just to have you see it.
These prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar. and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase, because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
If you like some of these visual ways to look at some of these processes that are kind of complicated, like replication and transcription and translation.
Kornberg wins the Nobel Prize for this. Good stuff.
Then there's a lot of them on YouTube. Has anybody ever done anything with the Khan Academy, or you're familiar with the Khan Academy? Khan Academy has a lot of tutorials on various biological processes. If you end up taking the MCAT, they partnered with the Khan Academy to provide studying materials for MCAT material, and so I ran across a lot of Khan Academy information. We'll show some videos when we talk about transcription and translation as well, but I would encourage you to check some of those out. So I think it's easier to see some things sometimes in terms of kind of three-dimensional processes instead of just the two-dimensional pictures you get in your book. One other issue that Chapter 13 talks about is some mechanisms that the cell has to repair damaged DNA. If DNA gets damaged, that can have some serious consequences. It can lead to mutation. There are a number of environmental factors that can damage DNA, and we're going to concentrate on just one of these right now. We'll come back and talk about some other environmental factors later on. But ultraviolet radiation can cause damage to DNA. You've all heard before, for example, that exposure to too much sunlight can increase your risk of skin cancer, right? Or if you visit tanning beds too much, that can increase your risk of skin cancer. Why? Because of the ultraviolet radiation that is present in the sunlight. Ultraviolet radiation is a shorter wavelength form of radiation than visible light. And the shorter wavelength that the shorter wavelength that light has, the more energy it has. And so ultraviolet radiation can be absorbed by skin cells, and it can damage DNA. Fortunately, your cells, if they're functioning properly, have forms of DNA polymerase that can repair the damage that occurs. And this particular type of damage is called excision repair. What do you do when you excise something? Brandon? You cut it out. Okay, you take it out. So excision repair is going to involve taking out these damaged sections of DNA and then replacing them with good sections of DNA. That's a different enzyme and a different process than mismatch repair. Remember, mismatch repair involves correcting errors that occurred during replication, kind of like the autocorrect on your word processor. This, once DNA has been replicated, if damage occurs to it, that damaged section can be cut out and replaced. So, what does UV radiation do to DNA? Okay, here's a normal segment of DNA with our two parental strands. If this DNA absorbs ultraviolet radiation, it can cause thymines in DNA to form unusual bonds. And those unusual bonds involve bonds between the thymine nucleotides on the same strand. And here is the bonding that occurs between these thymines. These bonds normally don't occur. Okay, they're not present in normal DNA. But those two particular thymines are not bonding with A's across from them on another strand. They're bonding with one another on the same strand. And that particular type of bond is called a thymine-thymine dimer. And thymine-thymine dimers aren't good for DNA. Because what they do when this new bond, this little one right here between thymines forms, is it distorts the DNA. And that can result in a problem for the DNA when it tries to replicate. So having this little bulge in the DNA because of the thymine-thymine dimer really interferes with DNA replication, and that's not good. So cells have some enzymes called nucleases which can cut out these thymine-thymine dimers, and then a form of DNA polymerase, remember there's 11 DNA polymerases, and one of the forms of DNA polymerase can then come in and fill in the required nucleotides to repair the DNA back to its normal state. DNA ligase is then needed to kind of seal everything up to make sure everything's bonded together properly again. So as long as this excision repair process occurs properly, then your cells have a pretty good ability to correct mistakes that have been caused by radiation damaging your DNA. Now, if you have too much DNA damaged, sometimes you can just overwhelm the ability of these enzymes to correct all the mistakes. So the more exposure to ultraviolet radiation you get, and the more dimers you form, the greater the likelihood that the cell won't be able to correct all of those. And they can lead to then changes in the DNA, mutation, And when we talk about cancer, you'll see that one of the first initiating events in cancer are mutation of genes. Okay, so be careful about your exposure to ultraviolet radiation. If you have defects in your DNA repair enzymes, then you can be very prone to certain types of skin cancers because your repair enzymes aren't correcting these mistakes that are forming in the DNA in response to that particular radiation. You were going to remind me about something at the end of last period.
Yeah.
You remember what it was?
So, the background error rate in evolution.
Oh, okay. Yeah, when we talked last time about the fact that DNA replication was really accurate and that there are all these enzymes that can help to correct mistakes that might be made during replication, and we said that the mutation rate was pretty small, about 1 in a billion, Dr. Morton came up to me after class and said we really should emphasize the fact that it's a good thing that that replication is not completely accurate, that it's not 0 out of a billion. You want to fill in just a little bit what you were saying?
You have 6 billion nucleotide bases in your DNA. Right? So if there's a 1 in a billion mutation rate, every time a cell replicates, there are 6 mistakes. Now, fortunately, most of those mistakes don't occur in coding regions and don't affect the phenotype. But without that variation, without that genetic sequence variation, natural selection has nothing to act on. And if you cannot adapt to environmental changes as part of natural selection, you die. So having a slightly error-prone DNA replication process is really important in the evolution of any organism on the face of the earth. That is one of the mechanisms by which genetic diversity is generated. Obviously, mutations such as what you see with UV can also play a role in that as well.
Yeah, we're going to talk later on after we kind of go through some more things about the central dogma, about the role that mutation plays in evolution. And actually in lab, you're going to see an interesting video that talks about mutation and the effect that mutation has had on the natural selection of organisms.
Well, just look around the room. We don't all look alike.
Right.
And we're thousands and thousands and thousands and thousands of generations away from 150,000 years ago when the first humans showed up in West Africa, East Africa. So there's been a lot of changes since then.
Okay, so when we began this, I said that the kind of topic of discussion that we're going to be working on for much of the rest of the semester has to do with this idea of the central dogma of DNA being transcribed into RNA and RNA being translated into protein. DNA kind of feeds back on itself in this central dogma because DNA has to be able to replicate. So we've spent some time now talking about the replication aspect of the central dogma. But what we need to dig into now more is this relationship between DNA, RNA, and protein. When we talk about this, really what we're talking about is a very important phenomena called gene expression. And basically what happens during the phenomena of gene expression is that instructions encoded into the DNA ultimately get turned into some kind of product. And that particular product then influences how the cell is organized, what the cell can do, particularly because of the enzymes that are controlling metabolism in cells. So proteins ultimately are the things that determine cell phenotype. Your DNA sequences make up your genotype. Last week when we started this, I said that there were two questions that were really very interesting about chromosomes and DNA. And one question was, how do chromosomes replicate themselves? So we spent some time going through DNA replication and understanding that particular phenomena. And understanding the structure of DNA was really critical in coming to the understanding of how DNA replicated. The more difficult question is, how does genotype determine phenotype? Does understanding the structure of DNA make you understand that fully? No. But it's a start. Okay? We have to understand the structure of DNA to answer this question. But it's more complicated, really, than replication was in one regard. So, how does genotype determine phenotype? I guess to answer that question, the first thing we have to do is we have to be able to define what that term gene means. Well, this is the definition that we're going to use for now of what a gene is. And this definition goes back to the mid-1940s. A couple of researchers by the name of Beadle and Tatum really gave us our first real working definition of a gene. I've used this definition before. It's a sequence of DNA nucleotides, and so it's A's, T's, G's, and C's. And this sequence of nucleotides is information. Just like computer code is information. And what's it information for? It is information for the primary structure of a polypeptide chain. Remember what primary structure is? It is the kind and order of amino acids. So if you want to make the alpha chain of hemoglobin, hemoglobin has 4 polypeptides, 2 alphas and 2 betas. And if you want to make the alpha chain so you can make more hemoglobin in your red blood cells, what do you have to do? You have to know the sequence of amino acids that make up that particular alpha chain. Because those sequence of amino acids dictate how that alpha chain folds up. And how the alpha chain folds up dictates how it interacts with the beta chain. And how the beta and alpha chains interact dictate binding oxygen and releasing oxygen appropriately. Okay? So how do we get from a set of encoded information to a functional product? If a gene has its information converted into a functional product, we say that that gene has been expressed. Okay? Are all genes expressed always? No. Think about your cells. We said last time that in your non-gamete cells, we call those somatic cells, your body cells, there are 23 pairs of chromosomes. 23 from mom and 23 from dad. And those chromosomes have multiple genes on them, bits of information that specify polypeptide chains. And the polypeptide chains that are produced when their encoded genes are expressed determine what the cells do and how they're organized. And not all of your cells are the same in your body. Muscle cells are different from liver cells. Do they produce the same proteins? No. That's why one's a muscle cell and one's a liver cell. Why? Because some cells are expressing some genes, other cells are expressing other genes. So sometimes genes are not expressed, the genes are turned off, and they're not converted into protein. In other cells, those same genes might be expressed, and they're turned on and make a particular protein. So one of the things after we talk about the central dogma and talk about how gene expression occurs is we're going to have to spend some time talking about the regulation of gene expression and what determines whether at a particular time in a particular cell a gene is turned on and expressed or whether it isn't. The regulation of gene expression is critical to the development of an organism. Okay, we all start out as a single cell. And eventually, as a result of cell division, we form an organism made of trillions of cells. But forming all the different parts of that organism's body and making livers and bones and muscles and brain involves gene expression being very precisely regulated, and at key times certain genes being turned on in certain cells but not in other cells. The more you learn about the development of an organism from a single cell to a fetus to an adult, and the gene expression that's involved in that process, the more you really get amazed that we all end up as normal as we do. Because even a tiny change in gene expression in one cell could really lead to horrible consequences in the outcome of an organism, in the development of an organism. So this is going to be really interesting information, but it's really complex information as well. We're just starting to really scratch the surface of understanding how gene expression is regulated and what turns on and what turns off genes. So, if this is our definition of a gene, genes are composed of just series of nucleotides, A, T's, G's, and C's. Proteins are made up of amino acids. So how does a sequence of these A, T's, G's, and C's ultimately get turned into something tangible, a polypeptide chain? That's really what the central dogma talks about. Your genes are where? In the nucleus, right? That's where your chromosomes are found, in your cells. But where does this process of translation occur? Happens in the cytoplasm, right? Associated with what structures? Ribosomes, right? So ribosomes in and of themselves are going to be really interesting. What are they made of? Do you remember? What are the molecular components, the two major molecular molecules that ribosomes are made of? Ribosomal RNA. What's the other part? Protein. RNA and protein. Okay. And they make up something that's really kind of interesting. They make up kind of a molecular machine. And the purpose of that molecular machine is going to be to string together amino acids and peptide bond them together to make this functional polypeptide chain. Where do those amino acids come from that we use to make protein? So Zach say food, and that's absolutely true. You eat other organisms, and in the digestion process, you break the peptide bonds, joining those amino acids together. They're transported through the blood and then moved into cells. And then you've got these amino acids inside your cells from the food you ate. With the help of the ribosomes and some other types of RNA we'll talk about later on, those amino acids are joined together via peptide bonds to make this functional protein that has a particular job in the cell. If it's a plant cell, maybe this is a peroxidase enzyme that detoxifies hydrogen peroxide. But how did it know which amino acid to stick next to each one? That information was encoded for a particular gene for that particular protein. So there's a peroxidase gene someplace in those plant cells' DNA. One of the things that kind of puzzled people when the central dogma was first figured out was, why this intermediate RNA step? Why don't we just directly turn the instructions into a product? Well, there are some advantages of having this RNA intermediate involved. And one of the advantages is really kind of a practical advantage. One of the analogies that's oftentimes made about instructions, DNA, is that it's a blueprint. Y'all know what a blueprint is? This represents kind of the master copy of information that the cell uses to make things it needs. is there an advantage to having these particular blueprints sequestered away within a nucleus? Well, yeah, there are. The master copy of instructions can kind of be protected there. And then if we then Xerox those instructions into a working copy, that can then move out into the cytoplasm and actually participate in the process of making a protein. We're hoping that we build and renovate this science building soon. And that's going to involve an architect making blueprints or plans for every aspect of this building. And there's a master set of plans that the architect has to develop. Do they want to take that one master set of plans and then hand it over to a plumbing contractor and say, use this set of plans to construct all of the pipes and HVAC and everything in this building? It's a little risky, isn't it? What if the plumbing contractor lost the plans? Or they got torn up? Or coffee got spilled out? Then we would lose that information. But by making copies, the architect can hold those plans in a safe and can make copies as needed, protecting that valuable information. During the construction process, we hope that we won't just have one group of contractors doing one thing in this building. While some plumbers and HVAC people are working on part of the building, we probably will have electricians doing wiring. Well, if there's only one set of plans, who gets them? Do the electricians have to wait for the architects? No, we make another set of copies for them as well. So the cell is going to have to make many different polypeptide chains, many different enzymes, for example. Think about all the enzymes that a cell has to make just to do glycolysis. So we could copy the gene for pyruvate dehydrogenase. We could copy the gene for phosphoglucoisomerase, all the enzymes required in glycolysis. And all those copies could move out into the cytoplasm, and each one of those copies could be translated on a ribosome, and so we could simultaneously make many different kinds of proteins. So this does make some intuitive sense when you start to dig into it and think about it a little bit. One of the things that I hope you start to understand about science is that there are always exceptions to rules. And the central dogma is an example of that. This particular concept says that DNA is always turned into RNA, and RNA is always turned into protein. Is that the way it always works? Almost always. But there are situations where RNA can be made into DNA. That particular process isn't called transcription. Transcription is DNA into RNA. RNA being copied into DNA is called reverse transcription. And that takes a special enzyme that most cells don't have called reverse transcriptase. There are some viruses that have RNA as their genetic information, not DNA. And some of these RNA viruses have this reverse transcriptase enzyme. And part of their life cycle involves converting RNA into DNA. When we finish gene expression, central dogma, cancer, one of the last topics we'll talk about is virus. And we'll come back and talk about this RNA viruses that have reverse transcriptase. They're called retroviruses or backwards viruses, RNA to DNA. Anybody know an example of a pretty notorious RNA retrovirus. The HIV virus is a retrovirus. So this is the norm, but there are exceptions to the rules. Oftentimes in genetics, when you find exceptions to the rules, they're happening within virus. So, here's a DNA molecule. In this particular representation, this particular DNA molecule contains 3 different genes. Gene 1, 2, and 3. Each one of those encodes genetic information for a different type of polypeptide chain. Each polypeptide chain will fold up differently, and each particular folded polypeptide chain will have a particular function. One of the functions might be, if this were the DNA of a jellyfish, the ability to produce this interesting protein called green fluorescent protein, which shines green when you shine UV light on it. Okay? One of the things we're really going to want you to understand when we work with green fluorescent protein is that it is the protein that is fluorescing. It is not the gene that is fluorescing. The gene contains the information how to make the protein, but only if that gene is expressed, turned into a functional GFP protein, will those bacterial cells glow green when you shine the UV light on them. If you shine the light on their DNA, they're not going to glow. That gene has got to be expressed first into a particular protein. What does expression involve? It involves a DNA strand that is part of this gene being copied into an RNA molecule, an mRNA molecule, and then this particular RNA molecule is going to be translated into a polypeptide chain with a particular sequence of amino acids. It's going to fold up in a particular fashion, and because of the way it folds, it will absorb ultraviolet light. Electrons will be excited, and as they drop back down to ground state, like they did in photosynthesis, what will they do? They'll release light in the form of that green glowing fluorescence. Okay? So, starting on Tuesday, what we need to do is we need to dig into this process of transcription. And then we might get into translation before the next exam, but I kind of doubt it. We'll probably just go as far as transcription. So Monday, what we're going to do is we're going to give you some introduction to the lab for the next week with the green fluorescent protein and transformation. And then you'll have your reading quiz over 13.1 and 13.2. Anything else that you want to say before they cut off? Nope. Okay. Have a good weekend.